Drawings of organic molecules. Organic molecules are generally more complex than inorganic molecules, such as CO2 and H2O. Different models have been created to illustrate them on paper. Molecular formula, the simplest, just shows the number of each atoms in the molecule. For example, C3H8. The condensed formula represents its physical structure in a simplified form, like CH3, CH2, CH3. Structural formula shows the physical structure in 2 or 3 dimensions, and skeletal formula is a simplified structural formula. These models provide a greater visualization of the molecule shape. Haworth projection, ball and stick model, which is a 3D configuration where the balls represent atoms and the sticks represent bonds, space filling, and ribbon. The condensed formula for a molecule captures its structure in a simplified form. For instance, the condensed formula for propane is shown above its structural formula in 2 dimensions. So here's the structural, here is the condensed. The connection between the structural and condensed formulas is shown above and to the right. This diagram shows all the atoms and bonds in the molecule, and it tells us a little bit about bond angles. Single bonds are represented by 1 line, double bonds by 2 lines, and triple bonds by 3 lines. While this type of drawing is often used for small molecules like glucose, it is too detailed for large molecules, and it does not tell us much about the shape of the molecule. Carbons are not always indicated on a structural diagram for large carbon-based molecules. So a carbon atom is implied at each vertex of the figure. Carbon atoms are often numbered in diagrams and referred to as C1, C2, etc. depending on their position. Some bonds are not shown. Common functional groups, such as hydroxyl groups, the C1 carbonyl group, and the C6 carboxyl group are shown without internal bonds. Skeletal structures are commonly used in organic chemistry. This is a simplified drawing style for organic compounds in which carbon and hydrogen atoms are implied but not drawn. This bond line structure appears as zigzag lines that represent single, double, or triple covalent bonds with 1, 2, or 3 parallel lines. These 2 representations are of the same molecule. Each corner, or at the end of the line, is a carbon atom and enough hydrogen atoms so carbon has 4 bonds. If present in the molecule, atoms other than carbon and hydrogen will always appear in the skeletal structure. It is optional to add certain functional groups. By adding solid and dashed wedges and wavy lines, structural models can be extended to 3 dimensions. A carbon atom is assumed to be at each vertex of the central figure. Single bonds are single lines, double bonds are double lines, and triple bonds are triple lines. When there are less than 4 bonds at a vertex, there are assumed to be enough hydrogen atoms to form 4 bonds with the carbon there. Wedge lines show a bond coming out of the page. Dashed lines show a bond going into the page. Wavy lines indicate that the direction of the bond is either unknown or a mixture of isomers. Given the following structural formula, draw the skeletal formula. And here it is. The carbons are attached at each corner and are connected by a straight line. Atoms other than carbon and hydrogen are explicitly shown. Hydrogens are not shown but are implied by the requirement for each carbon to have 4 bonds. It is optional to include CH3 instead of 3 hydrogens in the diagram. Given the following skeletal formula, draw the structural formula. Each vertex and endpoint of the terminal bonds represent a single carbon, identified with red dots, and its 4 surrounding bonds. Attach a carbon at each red dot. Keep the chlorine atom where it is. Then add enough hydrogens to each carbon to complete their 4 bonds. The molecule can also be represented by changing the zigzag carbons to a straight line, or by writing the terminal carbons as CH3 or H3C. All 3 of these representations are valid. Glucose does not usually exist as a linear molecule in watery environments. It exists as a cyclic structure shown below. In nature, C5 and the C1OH group are connected with a covalent bond. Click here to see the process. In Haworth projections, Wedge-shaped bond lines provide perspective. They indicate that this part of the molecule is projecting toward the viewer, out of the page. The numbers represent the carbon atoms. You will see the numbers again when DNA is discussed, 3' and 5'. Hydrogen atoms are not indicated, but are understood to be attached to the carbon atoms when there are not 4 bonds shown. The thicker lines provide perspective by showing that the atoms are closer to the viewer. Just as all of the carbon atoms are implied at vertices, hydrogen atoms bound to carbon are implied but not shown. There must be enough hydrogen atoms to fill the 4 vacancies of each carbon atom. How would this diagram look if every carbon and hydrogen were shown? Too complicated. The Haworth projection shows the shape of the molecule while not being too busy. There are carbons at each vertex and each forms 4 bonds. There must be 2 hydrogen atoms bound to C6, and carbons 1 through 5 each have 1 hydrogen atom bound. The projection can also be modified as viewed from slightly above the plane of the molecule, showing the parts that are above and below the average shape. The dark wedge can be used to show components that are out of the page. This model shows the 3-dimensional structure of a molecule by using balls to represent the atoms and sticks to represent the covalent bonds. Typically, carbons are black or gray, hydrogens are white, and other atoms have different colors. Oxygen is red, nitrogen is blue. The space-filling model diagram approximates the locations of electron clouds of the molecule and illustrates its shape. Here is the space-filling model of glucose. Here is the space-filling model of methane. Larger molecules require other diagramming methods that show the shape of the molecule without showing every atom and bond. This is a ribbon diagram of a protein that carries oxygen in blood, myoglobin. We see no atoms or bonds, only the shape of the protein in nature. The shape of the protein determines its function.